do this in person, but I am happy to present some of the things we've been thinking about for the better part of 5 or 6 years from now. And as of yesterday, this figure came out. I think it's quite pertinent. This is from Dustin Scheinhost's group at Yale, just kind of contextualizing the complexity of what we're talking about today in terms of predictive power of neuroimaging. And the huge component of this is, correction for bias, deciphering, signal from noise, biology from technical variation, et cetera. And I like to kind of think about this in a more formalized way in the context of my training in visual neuroscience, and calling this a kind of a prediction parallax. We've all been sitting in a car, you look outside, depending on where you're fixating in the landscape, things can be going in conflicting direction. And applied to kind of our prediction framework, oftentimes we take cross-sectional data points, even longitudinal, but in a constrained window of time. And to what extent, if we're doing this at different periods of development, is this more of a developmental outcome versus kind of a predictive metric to use going forward? I think different features across multimodal neuroimaging might be better suited to look at things that are more reflective versus predictive. And I'm just oversimplifying this into kind of categories of topography and topology, the idea of kind of emergent features after controlling for basic biological underpinnings that might have structural variation across population or across species, for example. And this, has really stemmed from the idea that most of what I look at, most of what I've been looking at in the context of brain networks has to do with this idea of covariation, which from the evolutionary biology perspective is defined by this notion of originating from variance generating processes. Without diversity in variance generating processes, you don't have covariations, another way to put that. Another humbling kind of, thing in the literature and just being kind of a student of neuroscience is this idea of assumption that things have been described before. This is a basic Google search of how big the human brain is. You'll get different, metrics, you'll get different, outcomes. You'll ask a different scientist, they'll give you a different feature that they're answering this question. And not to mention the fact that we can't really define variability. If you start to think about, well, what is the search space for possible brain size? It's about when you're looking, it's about who you're looking at, et cetera. And this is way more eloquently put in this model of embryogenesis, which kind of synthesizes this notion of emergent covariation amongst kind of overall large-scale trait variation. And what you're looking at here is an embryo developing in the first 8 weeks of life. And kind of the most fundamental feature of this is that the brain will emerge from basically nothing into something quite, substantial, in this case outlined eventually after the 30-second video in yellow. And not to mention the fact that, this all happens within an encoded space. Traits emerge, it's a finite, space. It's a quantifiable space. And I think that some of the most fundamental kind of features that I steal in some of the work that we do is emerging in the last few years. One of which on the left is complex figure, but using cell lineage specification and genomics, we can trace back things like anterior, posterior patterning in the brain into the first cell division. And looking at cell fate in the context of genomics has elucidated some very fundamental features that happen before the brain basically exists. And then we have this kind of obscuring, as I alluded to before, of time, where different transient and large scale processes interact in a spatially dependent way. And by looking later in development at patterns of covariation, what we're actually looking at are these intersecting remnants of temporal processes. And why is this important, especially in the context of brain networks? Well, we actually know a lot about spatial organization. We know that the relative position of different brain areas that are functionally important is encoded. It's conserved across species, et cetera, to some extent. We know that it's patterned just by virtue of some of the methods that have been applied to various connectivity constructs, notably kind of the gradient-based frameworks developed by Daniel Margulies a few years ago. We know that neurogenesis is patterned, which has to do with cytoarchitecture, which some Klaus's beautiful work over the last decade or so has elucidated the predictive validity in looking at cytoarchitectonic differentiation in the context of true anatomical connections. And we know from a practical side that spatial autocorrelation is incredibly important in the context of relating different brain maps. So this is kind of reiterating what I just talked about, which is why do we care? Well, size and related constructs are really important for overall organismal function, brain function included. Familiarity is often mistaken for understanding. We need to actually know whether the construct that we're looking at is a source of variation or whether it's a constraint on variation itself. This happens during development. It's very difficult to measure with the data that we have. And I think that we can leverage population-based inferences to inform different predictions at the individual level. So how do we map this? Well, we used one example of this in kind of past work, which is this very well-established statistical construct of allometry, which is anything but isometry. And this was first described in the context of gorilla femurs by Galileo a long time ago, noticing that the shape that accompanies increasing size variation of gorilla femurs, varies. And this is really, really fundamental. This was summarized by Gold et al. About a few decades ago, that like a larger organism must change in its shape in order to adapt to basically the environment. And so in the context of the brain, we know that across different constructs of variation, whether it's across species, across developmental time, and across individuals, after accounting for those two factors, we know the brain is variable. Across species is most notably kind of the biggest variability in overall brain size, just highlighting the greater than 100-fold variation across primates. We know that there's about a 2-fold variation accounting for development and evolution in the context of overall human brain size. And we know that even within the first year of life, the brain almost doubles in size. And so how can we leverage this size variation to look at overall cortical patterning? Well, David Van Essen's group in 2010, as well as us, kind of were the first to kind of frame this question in the context of local surface area expansion. And what we found was this kind of striking overlap, albeit some differences, in the local adaptation of surface area scaling in the context of changing size variation. In our case, we're looking across the population and modeling size across adult individuals. In Jason's paper in 2010, they were looking across developmental time in humans and across evolutions by proxy of looking at macaques versus adult humans. So how do these allometric effects play when we're looking at metrics of connectivity that most of us are interested in, DTI and function? Well, we typically are looking at subset of the species variation, and we're also looking at a handful of individuals typically. So that's one big restriction. But what the MAMI database has revealed from Asaf and colleagues is this really striking relationship that is pretty well established, that the white matter, which is the construct that we're measuring diffusion over, nicely scales with gray matter volume across the different species. But when you look at network properties, this starts to break down a little bit. And in the context of allometry, these R-squared values are actually really low. And so there's this notion that connectivity might have emerged in humans, or aspects of human-specific connectivity might be really important to overall function. This is kind of somewhat replicated, albeit in a different setting, and using slightly different metrics in a paper by Martijn van den Heuvel's group just recently, looking at other metrics from diffusion-weighted connectivity matrices across species and these allometric scaling relationships. Across other modalities, this hasn't really been done as much. In human fMRI, I found one paper that looked at different coefficients, graph theoretic properties, and overall brain size. And in some of Ben Folcher's work across species and mouse using the Allen Institute Gene Expression Database nicely showed that spatially autocorrelated expression patterns, which have a gradient-like pattern that is not dissimilar to functional patterns we see in humans, scale with brain size across mouse development. And just kind of synthesizing this using data that we've generated from a large-scale study that was published a couple months ago, along with the MAMI database, which Asaf and colleagues were nicely shared, we're starting to actually be able to unpack these allometric relationships in the context of Evo and Devo. So placing kind of this massive variation across age within the context of humans on the right here, in the context of overall evolutionary scaling patterns. And you start to see the incredible kind of power that you have when you're looking at scale and you have the data to be able to map these different constructs of the population. Now, these are really gross anatomical features that have very robust patterning structures in the context of the mean. Across a population, we can map overall scaling patterns, but the variation is still kind of an open question. But I do think that I hope it's a topic for discussion now, in that, if we have some of these simple features that we can readily extract across imaging, and we can map them and we can assess variation across age and across individuals, to what extent can we use that as a lens to look at questions about connectivity or topological patterns, individual variation, and features that we can extract from that in a normative model context that Enrico nicely kind of alluded to in his talk? How can we kind of work together in this way to better inform models of the human connectome, and especially in the context for prediction, whether it's behavior or disease onset, disease symptomatology? And so with that, I'll just thank my kind of current supervisors and previous collaborators, and would love to answer any questions and discuss this further.